It's a privilege to be here, and I'll say something that I really appreciate. One is that you're here. The second is that we have two discoverers of P53 in the audience, David and Arnie, and they're still leaders in the field. I think talking about aging, I think that's a tribute to aging. The third point is Fred Lee and Joe Fraumeni, the late Fred Lee, Joe Fraumeni, colleagues of mine at the NCI for many, many years, and obviously why we're here. And secondly, Joe, I think, changed the landscape of cancer epidemiology during his reign. So let me go on to aging and cancer and P53 isoforms. So one aspect of aging is called cellular senescence, and it's a normal process. It occurs either when there's damage to cells or developmental clues. They become senescent, and that means that they stop dividing, and they produce a number of cytokines that are important for attracting macrophages and for lymphocytes to come and recruit those. They recruit those cells in, and the macrophages phagocytize and kill, digest, and you have clearance, and then you have regeneration. This is all very good, except when it's persistent damage or pathology or aging, and there's senescence accumulation, there's chronic inflammation, there's fibrosis, and there can be cancer and premature aging diseases of various kinds. I mentioned that this is a normal process. It's a developmental process, and that's shown over here. And the easiest thing to think about is an embryo has all the fingers together, but senescent allows the fingers to be separated from each other. That's just one of many examples. The second point is damage-induced senescence can lead to a variety of neurodegenerative diseases that we study, including Alzheimer's and also cancer. So speaking of cancer, cancers can be benign. I should say tumors can be benign. And tumors that are benign generally have many senescent cells in them. And induction mechanisms include increased P16 and or P21. Latter is related primarily to P53. Free radicals, oxygen or nitrogen base, and telomere shortening that occurs with each division of the cell and uncapping of the proteins that protect. Cancers, on the other hand, have very few, in general, senescent cells. And there's what I'd like to call a barrier, a senescent barrier, that involves the P53 network and pathway and the RB network. But these can be bypassed. And one is P53 mutations, including when there's loss of heterozygosity, so the normal allele is also missing. But one question is whether or not Li-Fraumeni cells are also somewhat deficient in this senescent barrier. So cellular senescence, as I said, can be attributed to the telomere shortening, and you can see there is, as shown up here, a protein that binds to double-strand breaks will co-localize with the telomeres at the end of the chromosomes. DNA damage is another way. The DNA damage could be any place. And this goes through primarily the P53 and the RB pathway. And finally, oncogene activation, which causes a number of metabolic and free radical changes, goes through primarily, but not exclusively, the RB pathway. And there are various ways of detecting senescent cells, and I'll get into that in more detail in a moment. Now, P53 isoforms that J.C. and David detected quite a number of years ago, in 2005. This is actually a very important observation, but it's not a surprising one. And transcripts from 95% of multi-exon human genes are spliced in more than one way, and usually resulting in transcripts of varying expression with different cell types. So this is a very common event, either by RNA splicing or using different methionines. So a few years ago, actually more than a few years ago, we started investigating different isoforms. And we initially started out with investigating P53 beta that is missing some of the carboxy terminus due to RNA splicing, which we showed is due to a RNA splicing protein, SRSF3. And then there are two proteins, isoforms, that use different methionines. This is the second methionine, and this is the third methionine that lead to translation. And this one also has in this case, alternately spliced in. The delta 133 is controlled primarily by chaperone-mediated autophagy, so it's controlled at the protein level more than it's controlled at the RNA level. Now, why I was particularly interested in delta 133 is that the methionine start site is found only in primates and humans, not in dogs, not in rats, not in mice. So I thought that might have some particular significance, and that's partly why we've been investigating it. So we started out asking the question whether or not these isoforms are physiological regulators of replicating cell senescence. I'll just show you 2 or 3 slides that led to the initial results. And we actually studied something called cellular senescence or replicative senescence, in which cells keep dividing until they stop dividing and they become senescent. This was discovered quite a long time ago by Lynn Hayflick and Paul Moorhead. And you can detect these senescent cells by various senescent readouts. So this is a Western analysis, and really convinced us that this was something worth studying. And so these are 2 different human strains, fibroblasts, and they're either rapidly growing or they become senescent. And we found that in this Western that the P53 beta was detected only in the senescent cells, but not in the young growing cells. But the delta 133 P53 was found in the young cells, but not in the senescent cells. And delta 40 didn't change, so we actually stopped investigating that. So that was a clue. And we did, the initial experiment was to knock down the delta 133. I think that's much more relevant than overexpression kinds of experiments. And then ask, is there a phenotype? The phenotype we were looking for was senescent. And so this is just the knockdown. And there's already evidence that there might be senescence and activation of P53 by increase in P21. And the blue color here is senescence-associated beta-gal activity, which is really a measurement of lysosomes. And the knockdown of delta 133 increased this senescent marker, as well as a number of other ones. The other kind of question is whether or not the P53 beta will induce and the delta 133 will delay the onset of senescence. This is a very short-term assay, just asking the question about replication and cell proliferation, and taking cells that were near senescent, that's the vehicle control, overexpression of delta 133 seemed to grow faster. The P53 beta exposed cells grow much slower, and in fact, these P53 beta cells have undergone senescence, and there are a number of different senescence-associated markers that, including senescence-associated beta-gal, that are increased in the beta. Now, what was interesting to us was that the vector cells, which were very near senescence, they only had 3 or 4 more populations to divide before they underwent replicative senescence, were quite different than the delta 133 overexpressing cells, in which there was about the 10, 15 increased population doubling, but they were not immortalized. They stopped growing and became senescent after that period of time. So that was the initial kind of results. We published a number of papers that deal with various aspects. But since this is a session on metabolism and immunology, and Arnie's going to be talking about cancer immunotherapy, and we already heard some aspects of immunotherapy in the previous talk, I thought I would focus on this. Now, why we initially did these studies was not cancer immunotherapy. I was asked the question, is there evidence in vivo, in all of us in this room, of the switch between P53 beta and delta 133? And there was already a clue that this might be the case, and from some studies of lymphocytes. And these are the lymphocytes that are very important in cancer immunotherapy. And as they become less effective, people call them exhausted. And that actually we've shown that it's senescence what occurs in these. They were exhausted in terms of lacking of function for immunotherapy. So we took cells from lymphocytes from people of different ages, relatively young to relatively old, and we found that a co-stimulatory protein that's essential for these cells to divide decreases, and that's CD28. And if you do the opposite analysis of CD28 negative cells, you find that it increases with age. These cells that are CD28 negative have shorter telomeres. They have a number of markers of senescence, beta-gal activity, HP1 gamma, there's a heteroprotein accumulation. They have increased expression of various senescence-associated proteins in the phenotype, IL-6, IL-8, and so on and so forth. And they do have this switch in the P53s. So the cells that are exhausted or senescent have very little delta 133 and have high levels of P53 beta, as shown there. You could culture these cells in vitro, the ones that still have the replicative activity related to the co-stimulatory CD28, and they eventually will senesce and stop growing. And as they do that, CD28 positive cells goes down, and what goes up is the CD8 negative cells. So these senescent cells or exhausted cells have a number of different senescence-associated biomarkers, as shown here, including double-strand breaks that you can measure with gamma H2AX, and the amount of delta 133 actually decreases in these three different cells through three different patients that you culture in vitro. You can do the same experiment, the knockdown experiment of delta 133 in the lymphocytes, and they stop growing, and that's shown here, and they start producing a senescent-associated proteins and characteristics such as senescence-associated beta-gal, and the amount of the co-stimulatory CD28 also goes down. If you overexpress P53 beta, cells undergo senescence, they stop growing, and so this is reproduced. What we find in vivo is reproduced that's found in vitro. Now this takes us to the cancer immunotherapy part. So when we did these experiments, we examined not only senescence-associated beta-gal activity, but also IL-6 and IL-8 that are produced high levels in senescent cells, and PD-1, which you heard about just a moment ago, and LAG-3. These are both checkpoint proteins that are targeted in cancer immunotherapy. Now, we could find that this increase in these checkpoint proteins could be reversed by overexpression of delta 133, P53, in these lymphocytes that were senescent. And they accumulate, again, central memory proteins that are very important, and they decrease the amount of PD-1 and the amount of LAG-3. And they start growing again. Now, can one take this to actually samples from tumors and from people? So we examined 7 lung cancers and asked the question, in these lung cancers, what called TILs, that's T-cell infiltrating lymphocytes, or tumor infiltrating lymphocytes, what was the frequency of CD28? These are the effective CD8 cells, or the CD28 negative, which are the exhausted or senescent cells. And about a third of the cells in these tumors were actually the exhausted or senescent cells. And there's some evidence that they might actually be tumor promoting. So you want to have these CD28 positive cells. And you want to have CD8 positive cells because they divide, and you need more cells to kill more tumor cells. And it was shown that there are 2 actually co-stimulatory proteins, one of them being CD28. And this was described by a couple of papers that were in Science a few years after we published our paper, saying co-stimulation, a surprising connection to immunotherapy. Actually wasn't very surprising to us. So just to summarize this with the cartoon, proliferating and cytotoxic CD8 positive T cells have the co-stimulatory proteins, CD28. They have the memory proteins that if they undergo senescence, replicative senescence, that the delta 133 goes down, P53 beta goes up, they become senescent or exhausted CD8 T cells that produce the checkpoint proteins. They don't have CD28, and you can reverse this and reprogram back to these kinds of cells. And this has some implications in terms of cellular-based immunotherapy that we're pursuing. And I will make the point, including aging, people decrease the number of effective CD8 positive T cells if they have HIV or various virus infections, or they have cancer. And all of these lead to replication and replication senescence. So let's end up with this slide. So I've talked about 2 of the isoforms of P53 that we're particularly interested in, P53 beta. It's an orally spliced form, and delta 133 that is found only in humans and higher order primates. P53 is, as has been shown, is involved in almost everything, and it's certainly involved in senescence, and delta 133 inhibits it, and P53 beta enhances it. So P53-induced cellular senescence contributes to normal aging. I'm obvious one of that. And you find exhausted CD8 positive, CD28 negative T cells. And in the brain, reactive astrocytes are actually senescent. And with increased aging, there's increased number of reactive astrocytes, which we've studied. And that is associated with neurodegeneration and loss of neurons. Aging-associated diseases such as Alzheimer's, ALS, and HG progeria, which is a premature aging disease, they all have senescence as an important part of their process, And the astrocytes in Alzheimer's is that these are actually senescent and produce IL-6 that actually is neurotoxic to neurons, and these astrocytes stop producing nerve growth factor, which is neuroprotective. And we've published papers on each of those. We've just submitted a paper on late effects of cancer therapy to the brain and have found that radiation increases the number of astrocytes that become senescent, and that leads to a decrease in neurons and loss of cognitive activity. My training actually is in medical oncology, so I've seen that. Delta 133 can directly reprogram senescent cells to normal functional proliferating cells, and so far, without immortalization, and the experiments that we've done asking the question about oncogenicity have all been negative. Human embryonic stem cells and iPS cells have very high levels of Delta 133, much higher than normal cells. And we think that's partly related to the fact that Delta 133 actually is a dominant negative for P53, but only for genes that are involved in senescence, not in apoptosis. And we published that just recently. So one of the things we've been doing for the last couple of years, and it's going to continue for a period of time, is to investigate the hypothesis that increasing Delta 133 by small molecules or repurposed drugs is a therapeutic strategy for some of the things up above. So these are people who've done the investigation. These are the current people in the lab. Those are past workers. We've had wonderful collaborations with a number of investigators who've taught me a great deal, and I appreciate them, and I appreciate your attention today.
Time for a question, and David.
Sorry, just up here.
There you are.
Oh, yeah, sorry. So that's fascinating. George Williams wrote a paper in '57 called this concept of antagonistic pleiotropy, which seems to me really quite relevant, this idea of antagonism between tumor suppression and effective aging, which I guess, is it true that what you're driving at is trying to separate those two phenomena out so as to get long lifespans without cancer? Is that the payoff here?
No, my objective is actually improve cancer immunotherapy, including CAR T therapy.
Thank you.
The question about aging and cancer, that's a very different topic and has many different parts to it than cellular senescence and CD8 positive T cells. I think it's related, but that's just one small part of why I look like I do and you look like you do.
We both have beards. Very, very quick question, and then I'll do the last.
For LFS, does it mean that if you don't get cancer, since you have less senescent cells, it means you could be aging less fast?
The aging what?
That you, if you have LFS, that you would be aging less fast if you don't get cancer, but because you don't have that much senescent cells. Or is that not how it works?
A very interesting question. But I think the main part of aging is just replicative cells and also cancer for that matter. And for lymphocytes, you want to protect them from replicating and undergo replicative senescence. That's sort of the theme that I was trying to mention. And in cells like in progeria, in which these people who are 15 years of age look like they're 70, their Delta 133 is very low, and their CD8 positive T cells are primarily exhausted. So those two are related to aging and immunology. All right, thank you.